in this genome whose job is to encode for certain proteins that the frog uses to survive. We're going to cut this piece out using a restriction enzyme. This is the thing that Boyer discovered. It's kind of like a pair of scissors. By cutting the DNA at specific points, we can get different pieces. Now, there are tons of different kinds of restriction enzymes, and they all cut in different places in DNA. Our enzyme that we're using today is called EcoR1. Now, this piece of DNA has more than one spot that EcoR1 can cut, as shown by the dotted lines. So we're going to end up with other pieces of DNA in addition to the one we actually want. Now, that's actually okay, because we're going to deal with that problem later. What's going on in the tube is we're literally just cutting up our piece of DNA, just like this. Now, here's a tube full of plasmid. It's basically a circular ring of DNA that can exist in bacteria, and it basically encodes for certain traits that affect how bacteria look or act or survive. Now, to add our DNA from the frog DNA to our bacterial DNA, we need to cut the plasmid open too. So we've got to add our EcoR1 scissors, and they're going to cut at this site. You let this reaction proceed, letting all the ingredients, the plasmids, the restriction enzyme, and some water and buffers to hang out together until all the plasmids are cut like this. Now we need to add the DNA pieces from the frog and allow the pieces to... You let this reaction proceed, letting all the ingredients, the plasmids, the restriction enzyme, and some water and buffers to hang out together until all the plasmids are cut like this. Now we need to add the DNA pieces from the frog and allow the pieces to join our cut plasmids. Here's what's going on in the tube now. I've got pieces of frog DNA, and I've got cut plasmids like this. I need to add this guy into this plasmid, and the way I'm going to do that is by adding an enzyme called DNA ligase. This enzyme kind of acts like tape, and it fixes the DNA insert into the plasmid. Now, another important thing about the plasmid itself is that it carries a marker that makes it resistant to a certain kind of antibiotic called tetracycline. This will come in handy in a little bit. After a little while, we'll have a couple types of DNA going on. This is the one that we want. It's plasmid, and it has the purple frog DNA insert. But we'll also get things like this. We'll get plasmids that had other parts of the frog's genome inserted into it. And we'll also get plasmids that just close back up on themselves. So now that we have these new plasmids, we've got to stick them into some bacteria. Here we're going to take some E. coli. As you can see, it already has some DNA in it. The important thing about these guys is that they'll actually die if you add the tetracycline antibiotic, which we talked about earlier, to them. No. What Boyer and Cohen did to get the plasmids inside of these E. coli is that they cooled all the cells down and then quickly heated them up, creating little holes in the cell membrane. Then the plasmids could slip through and get into the cell. These are the kind of bacteria that we have now. Bacteria that got the plasmid we wanted. These are the guys we want. But we'll also get bacteria that got the plasmids that we didn't want, say this one with the wrong piece of DNA, or this one with just a plain plasmid without any frog DNA. And more than likely we'll get ones that just didn't pick up any of the plasmids. Now what I'm going to do next is plate these cells on some tetracycline agar plates, which is basically some fancy jello that has antibiotic in it. So these guys won't be able to grow, but these guys will. I've plated the E. coli that we transformed with our new plasmids onto this plate, which has tetracycline in it. We're going to let this sit in the incubator overnight and allow our bacteria to grow, and we'll check on it again in the morning. Alright, so as you can tell, we've definitely got some colonies that grew on our plate overnight. So now the problem that we have is we need to be able to distinguish the bacteria that got these plasmids from the ones that got these plasmids, which don't have our frog DNA in them. So what we're going to do is use the same techniques that we did at the very beginning of this experiment.